All right, guys, welcome back to our ongoing discussion of energy extraction via glycolysis. So, if you think about everything we've talked about up to this point, right? So, we have glucose. This was our starting point. So, what then, if you think about running glycolysis, those 10 steps of this catabolic breakdown we've talked about, right? Um, what are the products up to this point? Right. Well, we know one of the primary products, right, is what we get out the back end of this pathway, which is of course pyruvate, right? That's that 3 carbon um, uh, alpha keto acid that is the essentially glucose split in half once we've run the set of reactions, right? And we know that a big point of this whole pathway, right, of course, um, so we get pyruvate as one of the products, right? And we know a big point of this pathway, of course, is also to make ATP, right? So we're taking ADP and we're making ATP through those substrate level phosphorylation events, right? So ATP is another one of our products. So pyruvate, ATP, there's something else if you think back. There's one other primary product of this pathway. It's really important. It's a key step right about the halfway point, right? And without it, we're not even set up to do those substrate level phosphorylation events to make ATP. Remember, it's this key oxidative step where we take NAD plus and we make NADH, right? So NADH is one of the other key products of the glycolytic pathway, right? And there are a couple of really important points here. If you recall, right, the enzyme that runs that reaction is, of course, GAPDH, glyceraldehyde 3-phosphate dehydrogenase, right? And GAPDH, to function, GAPDH has to have NAD, right? It's an essential cofactor. So another way to state this is that if you don't have enough NAD plus around, right? If you don't have enough NAD plus around, the GAPDH reaction won't work. And without the GAPDH reaction, glycolysis is dead, right? You don't even get to the point of recovering the energy investment phase without GAPDH, right? So really key idea here is that the available pools of NAD plus, the oxidizing equivalents available in NAD plus have to be maintained, right? If you don't have that molecule, you don't have NAD plus, you got a problem, right? So there has to be then some way, or an important aspect of the subsequent part of this pathway is we have to have some way to regenerate that NAD plus by extracting those high potential electrons, right? To recreate the NAD plus, right? Now, under most cellular conditions for organisms like us, the need to regenerate that NAD plus pool and dump these high potential electrons are co-aligned, right? Because if one has oxygen available, dumping those high potential electrons into oxygen and consequently making a whole bunch of ATP, right, are sort of two things that are aimed in the same direction, right? We can get rid of we can regenerate our NAD plus pool, and we make a whole crap ton of ATP in the process, right? We get, and let's just keep the GAPDH reaction running, right? So here's the question: what happens when you don't have any oxygen, right? And this isn't just an issue for microorganisms growing under conditions where there's no oxygen available, right? Of which there are lots and lots and lots of organisms, right, that grow under so-called anaerobic conditions, right? What do, right? You got the same basic problem. You still got to run glycolysis. You don't have any oxygen. What do? Another way to put this is that a really important thing that cells have to think about, to maintain, is this ratio, this NAD plus to NADH ratio is a super important sort of cellular condition. Cells are going to work very, very hard to maintain that ratio so that there is a good balance between NAD plus and NADH, so that NAD plus is available not just for the GAPDH reaction, but other really important reactions. It's just we're super focused on this one really important one right at the top of intermediary metabolism. Okay, so if cells need to maintain that ratio, even under conditions where there's no oxygen available to dump the electrons, what do? Okay, well, the answer to this is you give up on this idea of using those electrons and their potential energy to synthesize as much ATP as you can, and you basically turn around and think about them as chemical waste, right? Right. You got to get rid of them. You got to um, uh, pull them out of the structure of glucose in order to get down here to the second half of the glycolytic pathway and make some ATP. But once you've done that, you got to regenerate that NAD plus pool. So you basically have to just like bail and recreate that NAD plus pool, right? The reactions that are responsible for doing this in the absence of oxygen that allow glycolysis to continue running are known as fermentation reactions, right? There are a variety of different ways in which cells under anaerobic conditions manage to solve this particular problem of keeping their NAD plus to NADH ratio in an optimal set of conditions by dumping these electrons that come flowing out of reactions like the GAPDH reaction. We're just going to look at a couple of them, right? We're going to look at two examples of fermentation reactions. One is a reaction that's super relevant for our own physiology, so-called lactic acid, also known as homolactic fermentation. The other is so-called alcoholic fermentation. Okay, so let's take a look at this this homolactic pathway first, right? It's pretty simple, right? There's essentially a single step to make this happen. All right, so the homolactic fermentation looks something like this. Remember, the goal here is we got to do something with those electrons in NADH, right? So one really simple solution is to say, well, this was one of the products of glycolysis to this point, pyruvate, right? In principle, there's a really good place to stuff some electrons, and that's in that carbonyl right there. So that's more or less what homolactic fermentation does. It takes pyruvate, takes that ketone group, takes the electrons that were generated by the GAPDH reaction and simply stuffs them back into that molecule to create this carboxylic acid containing alcohol called lactic acid, which then regenerates the NAD plus pool. The enzyme that runs this reaction is, of course, lactate dehydrogenase. or LDH for short, right? Interesting. Why do you think it's called um, lactate dehydrogenase? Because it's not running as a dehydrogenase the way I've described it here. It's running as a reductase, right? It's taking NADH, is reducing that carbonyl group, and it's turning it into an alcohol, right? Well, the reason is kind of like with pyruvate kinase and many of the other enzymes that have their names fixed by virtue of how they were discovered. So this enzyme was originally identified as running in this direction, which, like so many all enzyme, you provide enough the the products, you can drive the reaction in the opposite direction. And lactate dehydrogenase will run oxidatively, but for the purposes of for the fermentation process in tissues, where this is important, the enzyme runs in the opposite direction. I don't know the free energy balance off the top of my head, but it's a pretty favorable reaction. If your NADH concentrations and your pyruvate concentrations are sufficiently high, LDH will quite happily make lactic acid, right? And the LDH, this enzyme, is present in most metabolically active tissues in organisms like us. And it's super important in high metabolic tissues, for example, like muscle, right? And the reason for that is that under very high metabolic load, it's not possible to get sufficient amount of oxygen into those tissues to maintain the other major pathway we're going to be talking about that comes off the end of glycolysis, where you get your maximum ATP yield. You just can't get oxygen in there fast enough to run it. And so those tissues default to running glycolysis. And to keep running, they have to do something with those electrons. And so LDH becomes super important to keeping that reaction going, generating lactic acid, right, and NAD+, so glycolysis keep running. Now, under normal conditions, again, in organisms like us, there's a way to recover all that energy that's still available in the structure of that lactic acid, right? That thing's not fully reduced. Remember, fully oxidized. The fully oxidized endpoint is carbon dioxide, and there's still quite a bit of good free energy in that lactic acid. And as we'll see when we talk a little bit later about the Cori cycle, organisms with more sophisticated metabolisms and tissues like us can offload that recovery process by exporting this lactic acid into the bloodstream, right? And essentially letting the liver pick up the slack, right? We'll talk about this, like I said, a little bit more later when we talk about the Cori cycle. But one interesting issue is for anybody who's ever overdone it, doing some exercise or working out in the gym, they've experienced the consequences of lactic acid buildup in a pretty big way, right? Because if you are unprepared in your skeletal muscle tissue to dump this lactic acid back to the liver, basically your tissue's not sufficiently vascularized, what happens is the lactic acid builds up in those tissues in your muscles. You sort of overdo it in the gym or whatever, right? And that consequence of that is that the pH in your muscle tissues drop way too fast. And that drop in pH causes all kinds of unfortunate consequences in those tissues, which results in the horrible soreness that anybody who's ever overdone it in doing physical exercise has experienced, right? Okay, so that's the fermentation reaction. And again, like I said, we're going to come back to this later. It's going to be important for organisms like us. And it's the main fermentation reaction that we would care about for, say, humans or mammalian systems. But there's another really important fermentation reaction that's worth knowing about, partly because the chemistry is kind of cool, partly because it has all kinds of important consequences for us on a Friday or a Saturday night, and that's alcoholic fermentation, right? Okay, so this is not a fermentation reaction, like I said, that happens in organisms like us. In fact, it's not a fermentation reaction that happens in even most microorganisms, but it's a type of fermentation reaction. It's going to take 2 steps to get this to happen that certain strains of fungi, unicellular fungi, yeast, have gotten really, really good at. And even more, our ancestors got really, really good at manipulating the yeast to get them to do this, right? And that is essentially to solve this same problem by creating an alcohol, right? And so it's worth sort of asking the question, why do yeast do this? I mean, why do they solve the problem this way? Why not just make lactic acid, right? Well, again, part of the answer to this is they've got the same basic problem. If yeast are growing anaerobically, they've got to solve the same issue with regenerating their NAD plus pool to keep glycolysis running, right? But yeast have turned that issue to their advantage in a really interesting way, where they've basically come up with a strategy for creating a metabolic waste product that gives them a selective advantage under certain conditions, right? When yeast are growing anaerobically, and there's enough glucose available, or there's a large glucose a carbon source available, on glucose, right? Yeast can secrete, or certain strains of yeast can secrete really large amounts of ethanol, right? It can be upwards greater than 12% culture volume to volume ethanol as a consequence of this pathway, which is something like 2.5 molar ethanol, right? The thing that yeast have figured out is that they can survive in 12% ethanol. Very few of the other organisms they might be competing with under those conditions to utilize that glucose, very few other organisms can live in anything greater than about 5% ethanol. So what yeast basically have figured out how to do is to metabolize the crap out of the available glucose, kill everybody else off in the environment that they're competing with them, because essentially they're secreting an antiseptic, and then they can slowly, maybe if they get so a little access to some oxygen, they can then happily metabolize that ethanol at their leisure once everybody else has been wiped out, right? Which is kind of a cool idea, right? All right. So how does alcoholic fermentation work? Like I said, this has got 2 reactions, and the first reaction of alcoholic fermentation involves some interesting chemistry that's going to be important, or is important in other similar chemistry happens in other pathways in intermediary metabolism in organisms even like us. Not this pathway, but the cofactor that we're going to use here, and what it's doing. That's why I'm illustrating it, is similar. So it's important in terms of the basic chemistry. So again, we're going to take pyruvate, right? And what we're going to do is we're going to drive a decarboxylation reaction. We're going to take pyruvate and we're going to drive off that CO2 group here, right? And we're going to turn this into an aldehyde, specifically acid aldehyde. The enzyme that runs this reaction is known as pyruvate decarboxylase. Right? And if you look at that and you think about it for a second, you're sort of like, wait, how does it do that? Right? Give that a little thought. You're like, well, let's write this out a slightly different way. If you imagine the chemistry that might be going on here, we've got something like this, right? And we need to split that bond. So you're like, okay, well, fair enough. I could push those electrons there, which would, what, split this bond here? But then, wait a minute, I've got to generate something that looks, in terms of as a sort of a transition state, that's like the carbonyl with a carbanion on the carbon. How favorable do you think that's going to be? It's not, right? So spontaneous decarboxylation of an alpha keto acid like this just isn't going to happen, because that transition state is so energetically unfavorable, it's just not going to happen. So pyruvate carboxylase, to get this reaction to go, but I said it happens, right? It's got a really important cofactor. And the important cofactor is known as thymine pyrophosphate. Or TPP, right? And TPP has this really interesting characteristic, right? The way this cofactor works, right? So TPP looks something like this. All right. So there's this amino pyrimidine ring, right? On one side of the molecule. On the other side of the molecule, it's got this sort of diphosphate, this phosphodiester, a couple of methylene groups. But the really key business end of this molecule is this thiazolium ring right here. And the reason this is really key is because that carbon right here between the nitrogen, that quaternary nitrogen, right, and that sulfur, that proton on that carbon is incredibly acidic compared to a normal carbon-hydrogen bond, right? And when that proton pops off, right, you can generate a extremely strong carbanion, which is a great nucleophile, number one, and that carbanion is stabilized by resonance in the rest of the structure, right? And that becomes important because we're going to use this molecule to do some covalent catalysis to help drive down the free energy of that that carbanion like intermediate that we have to form in order to decarboxylate pyruvate, right? So let's see what that looks like. All right, so here's TPP. Here's that diazolium ring, that business end. There's that acidic proton. When it deprotonates, you get something known as an ylide, right? Which is this sort of same basic structure, but with that stabilized carbanion on it, right? Like I said, that's a really good nucleophile. And if you drive this with a little base catalysis here to populate that carbanion, it will then attack in the active site of the enzyme pyruvate that is bound in just the right orientation, and it will go right after that carbonyl carbon, giving us then a sort of an intermediate state that, you know, we push those electrons into the oxygen to form an alcohol group. Now, decarboxylation of the pyruvate is massively more favorable, because if we now pull those electrons over here, split that bond, we now have a resonance, a nice deep resonance into that positively charged nitrogen on that thiazolium ring that can stabilize that nascent carbanion. So remember, if these electrons came back down, we would normally have to stabilize our carbanion right here on this carbon that's also in this carbonyl or alcohol carbonyl group. Super unfavorable. But now we have another place to do that. We bring these electrons down. We have this nice carbon nitrogen imine like double bond. It's a quaternary amine, or the quaternary amine like molecule, positively charged. Great place to stuff some electrons. Really easy to now make the electrons flow here to form this sort of stabilized, resonance stabilized carbanion. Essentially, that carbanion is still kind of there, right? You can think about it as being there, but the resonance stabilization provided by the TPP makes the free energy of populating that state dramatically lower, right? And so here again is another example of where that covalent catalysis lets you do something that's just not going to happen in the absence of the covalent adduct that provides that stabilization, right? Okay, so now all we got to do is sort of reverse this process, right? Or push it a little bit further along. We now deprotonate, right, that that alcohol, because remember, we're going to make an aldehyde. So we got to pull that proton back off. A little base catalysis here, assist, will drive those electrons down into that, recreate that carbonyl. We now split this bond, which now pushes the regenerates the ylid form of TPP, which can again be reprotonated to form TPP in the active site of the enzyme and release acetaldehyde, right? Okay, so that's basically the strategy you would need to use, something like that, in order to decarboxylate an alpha keto acid, right? Okay, what is the second step of alcoholic fermentation? Well, it's actually pretty easy because that was the hard part. So reaction 2 of alcoholic fermentation is just to now take that acetaldehyde right? So we decarboxylated pyruvate, made acetaldehyde. You can look at that and think, well, that's not too far away from where I need to go, because all I need to do now is dump that NADH into that structure, right? Reduce that carbonyl to an alcohol group, right? And at this point, I will have generated ethanol. Right? The enzyme that runs this reaction, of course, is one that we're all familiar with, alcohol dehydrogenase. Right? And when running fermentatively, this is the direction the enzyme is going to run. It's going to take that acetaldehyde, right, the NADH, and create ethanol. Right? Cool. And that's the ethanol that yeast then export, whatever, right? Interesting variation on this is that we, humans, have a version of this enzyme in our livers, as we all know, on that Friday or Saturday night, when we take the products of this fermentation reaction so generously provided by the yeast, and we basically now need to do something with this alcohol in our livers, take it and turn it into acetaldehyde. All right, as your body, your liver works to detoxify the alcohol from a glass of wine or from beer or whatever. And a little too much of this relative to the rate of subsequent metabolism in the liver, buildup of acetaldehyde is the thing that gives you that really awesome feeling when you've had a little too much of this on that Friday or Saturday night, right? So, okay. All right. So that more or less brings us about as far as I want to go into these fermentation reactions. So let's just sort of some final thoughts, a little accounting, right? If you think about this. Okay, so the fermentations solve our NAD plus NADH balance problem, right? Okay, but it's when you think about it, it's a really inefficient use of glucose, right? So glycolysis plus fermentation has a net ATP yield of about 2 ATP per glucose. Glycolysis plus the subsequent pathways we're going to talk about later, those oxygen-dependent pathways, as you'll see, can generate 38 ATP per glucose, right? So glycolysis plus fermentation, right? That's an effective energy yield of, in the range of 5%, by any measure, that sucks, right? I mean, that's an incredibly inefficient use of the energy available in the structure of glucose. 5%. So why do it? All right? Why do it? Why do this? Given that fermentations are so inefficient, right? Well, one reason is you have no choice. Under some circumstances, you have no oxygen. You want to continue to live, or you continue to run those deep tissues you can't get any oxygen to. 5% is better than zero, right? So either because you're a microorganism who has no choice, there's no oxygen, this is the best you can do, and you're just going to live with it, or because you're a tissue that you can't get oxygen to right now, some ATP is better than no ATP. And if it's inefficient, well, that's just the price, right? And in a lot of cases, like in organisms like us, where there is an ability to recover that remaining available energy by using sort of offloading the recovery to other tissues, it's not such a big deal, especially when you've got plenty of sources of glucose being made by those other tissues, right? It makes a lot of sense to do it this way because the long-term consequences are not serious, right? There's another important reason why cells would do this, and it has to do with speed. Yes, these oxygen dependent pathways are dramatically more efficient, make much more ATP per glucose, but they're also relatively slow. By comparison, the glycolytic production of ATP can basically run at about 100 times the rate at which those oxygen dependent pathways can produce ATP. So if you really need to make ATP fast, glycolysis is the way you go. Those oxygen-dependent pathways, they're more efficient, but they're a lot slower. And there are times when the most important thing in the world is to make as much ATP as you realistically can, as fast as you realistically can. And under those conditions, glycolysis is the pathway to choose, right? There's a third reason, and it has everything to do with the intermediate species that are made as we went down the glycolytic pathway. There are things that are created there that get used for making other biomolecules, right? This is an example of reactions that are sometimes called anaplerotic reactions, right? Where the pathway is run for the purposes of creating intermediates that are needed for biomass, right? And we're going to see an example of that aspect of running glycolysis rather than thinking about it as an ATP producing pathway in the paper we're going to read for next time.